Okay, thank you for being here. Did this. Make a new notebook. I want biology. Okay. Okay. Now biology. As you know, biology can be divided as living organism. Study of biology basically means living organism. We study here, and formation and formation slash basic unit of structural unit. And biology as living organism can divided by cells. Cell can be of two types: multicellular and unicellular. As unicellular examples are bacteria, they have only one cell. Another is amoeba. Another is yeast, which is to make curd. Multicellular is multicellular. It could be like your human beings or like dogs and animals and all that. That is multicellular. Now, going on the basic, like who created what. So, 1665, Robert Hooke. Robert Hooke. Uh, he saw like the cell of a leaf from a magnifying glass, and what he saw was a honeycomb structure, something like this honeycomb structure he saw. And when he saw this, he got to know how cell our structure is formed. What is like the cell structure that is done by Robert Hooke in 1665 as honeycomb structure. Next, 1674, Antonio Leeuwenhoek. Antonio Leeuwenhoek. He took a pond water and saw bacteria in there. So you could remember as Leeuwenhoek. Hook is saying hook is basically is the stuff which you use for fishing. So fishing would be done in water body. So you could associate Antonio Hook with that. And 1967 as 67. Now one more cell theory of two brothers, which is 1838 and 39, which is done by Sheldon and Szechuan. They said a pretty important stuff. They said that all living organisms are made up of pre-existing cells. Like, supposedly your grandmother is there. She had a baby, which is your father. Now, with your father and mother, you were born. So basically, the cell is never being created. It is just getting transferred from one body to another. Like that's what Seinstein and Schwarzbaum said in 1838 and 39. Now, going on ahead, uh, 1855, Rudolf Viscoch. Rudolf Viscoch said that all new cells will arrive from pre-existing cells, and they said the cell is fundamental unit of life. Rudolf Viscoch. Smallest cell is micro ना overall human में है sperm male and female gamete. Okay, now going on ahead, how do I do this? Okay, next is difference between prokaryotic and eukaryotic. So prokaryotic are just in their name is there is pro, but they are actually no. This is a trick that I know that I used to learn it. So pro is there, but they are actually noob. Noob in a sense, they don't have no nucleus. No nucleus, which is actually well developed. Next is they have cell membrane. Next is they have cytoplasm because cytoplasm is important as it provide like storing capacity, and you are can't store everything in air. You need some liquid that can protect and help in transferation and all that. Liquid like structure cytoplasm. Then they have genetic material, as cells need to have genetic material, and but they have no nucleus membrane. This is important. Prokaryotics don't have nucleus membrane. Next, they have cell wall, but it is for only some prokaryotic have it. Only some, not all have that. They are unicellular. They have single chromosome. Then they are small in size. Then they have no membrane. Now going on to your eukaryotic cells. Eukaryotic have nucleus which is centralized. Eukaryotic have a paired chromosome. which are 23 of pair. Then they have large cell body, eukaryotic. Then they are multicellular. Then they can be unicellular, which is amoeba. Okay, now this is written in my notes. Endoplasmic reticulum for Golgi bodies. So endoplasmic reticulum is secreted from the Golgi endoplasmic reticulum secrete Golgi bodies. Endoplasmic reticulum protein production Golgi bodies and then transfer. Endoplasmic reticulum. Endo. Then here. Then cell membrane, an outer layer of cell membrane. An outer layer of cell membrane, extra rigidity and hardness is provided. So cell membrane, it provides rigidity and hardness. Now cell membrane in different things are different. So in plants, cell membrane is called cellulose. In fungi is called chitin. In bacteria is called peptidoglycan. And cell membrane is semi-permeable, as stuff can go through cell membrane. It's just to provide extra rigidity and hardness. Going on ahead. This is hypertonic solution, isotonic solution, and then high oh hypotonic solution. Hypertonic solution basically means the solute, which is supposedly the thickened stuff. You know how there's a juice which is like thick and you put it in a cup and then you put water on it, it is concentrated. So when you put water, it become dilute, so it expands, right? So same logic is following here. Hypertonic is saying that if the solute is more and the solvent, which is water, is less, then what will happen is, this is the plant, right? So inside of this, the solute is more and the solvent is less. So if there is thickened stuff at the below, which is thick, and you put water on it, so what happens? It expands, right? So here also this would expand. And in plant, it call, in animal cell, it shrinks. And plant cell, it is called plasmoides. Plasmodiasis for a hypertonic solution. So a question which is have been asked is, a cell will undergo plasmoids if it is in which solution? A cell will go plasmodiasis if it is in which solution? So here it is a PYQ and its answer is your hypertonic solution. Now going on to isotonic. Isotonic is normal. Basically the solute and solvent both are equal. So nothing happens to the center. Now your hypotonic solution is when the solute is less and solvent is more, then it would blow up. No, in like if it is, so this will swell up in animal cell, and in plant cell it increase turgor pressure. In a hypertonic solution, what would happen is it will shrink. So the shrinkness is something like, like how there is a low high amount of solute here, right? So what would happen, the solvent outside is low, so what would happen, the solute would try to go outside, and when it is going outside it would shrink to resin kind of thing, right? You are basically taking out the nutrients. Here it is like the solute is low and solvent is more, so it will go inside it here and it would swell up. That's what it is. And the question is, the a cell will go plasmoides if it is under which solution? So hypotonic solution. Now a petty clearation. Things which have protein in it are your nucleus, nuclei, cytoplasm, mitochondria, chloroplast, and ribosome, which is actually the production of protein, of endoplasmic reticulum. Going on, nucleus. Nucleus is the brain, and nucleus was discovered in 1831 by Robert Hooke. Sorry, Robert Brown. What I'm saying, Robert Hooke is the cell one, 1556. Now, nucleus, I have to draw. This is cytoplasm. This is the double membrane for nucleus, which is only in eukaryotic cells. And this is inside of nucleus. So this fluid is called as nucleoplasm, and this is nuclear membrane, and the thread-like structure is chromatin. Chromatin, which have DNA plus protein. Chromatin, DNA plus protein, and middle is nucleus, which have RNA plus ribosome, protein factory. Now, there are difference in RNA and DNA. DNA is ribonucleic acid. RNA is ribonucleic acid. So, just a thing that you need to remember is what I usually do is RNA only have something different from DNA, which is uracil, and DNA, which have different from RNA, is thymine. So DNA is thymine, which is singular in like it's different than RNA, and goes for RNA, it is uracil. Now, nucleoid, undefined nucleus region in prokaryotes. Now endoplasmic reticulum discovered by Keith Potter endoplasmic reticulum. Uh, endoplasmic reticulum are in tubules form tubules or cisterns form and found near as well as from nucleus. Now rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum have ribosome, which are the protein synthesis one. Ribosome, protein synthesis. And smooth endoplasmic reticulum is for vertebrates uh, and capture and remove toxin, detoxify. And smooth doesn't have ribosome, obviously. Smooth ribosome main function is synthesis of fat and lipid. Then mitochondria. Mitochondria. First of all, basic definition: powerhouse of the cell. And he does this thing by uh, producing ATP. ATP is produced, which is ATP also is a currency of energy, which ATP's full form is adenose diphosphate. So what happens is like at the end there is three kind of chain here. So if you have to burn energy, it will remove one, then second, then third. That's how it burns the energy, and that's why ATP is called the energy currency. Now helps in circulation. Mitochondria also helps in cellular respiration, which is done by the ATP. And going on ahead, like plastids are not found in animal cell, as it is for like storing and all that, and it is very enormous in plant cells, as it is like prominent in like major 80% of the space has been taken by plastid in plant cells. So it is not in animal cell. If it is, then it is in small, very like small size and all that. And this is done by Ernest Haeckel's discovery in 1866. Now, uh, this plastid is double membrane and semi-autonomous. Now, plastids are of different types. First on plant, chloroplast, which is green. Now, the colored pigments are first of all caxtoin, and xanthophyll. Xanthophyll is yellow, caxtophil is red or orange, you could say. You could remember as cactus, you got hit with it, you become red and yellow. With xanthophyll, it is yellow only. Sorry, orange and red. Now, this is important. Leucoplast is colorless, which helps in storing starch. And this is done by amyloplast. So amyloplast is responsible for storing starch in plant cell. Amyloplast. And oil is done by alleloplast and protein is done by proteinoplast. Okay. And chloroplast known as kitchen house of the cell. That is basic knowledge in my opinion. Then Golgi apparatus, which are like tubules and all that. This kind of structure is there, which have crystals in there, crystals, and they are pouch-like vessels. Function of Golgi body is transportation, packaging, and synthesis. Making lysosome also is the Golgi apparatus makes lysosome too. And yeah, then double membrane, then this is different stuff. Now some basic clearation I have done here. Double membrane people, first is nucleus, as it has the normal membrane and the nucleic membrane in only what is called ecloplast. Nucleus protein histosine are highly alkaline protein that package DNA into structure unit called nucleosome, and it has histones. Highly alkaline they are. न्यूक्लियोसोम बनाते हैं ये nucleosomes. Then mitochondria, then plastids, as plastid is like chloroplast, and these all have protein inside of it as we have done earlier. Now single membrane, which are endoplasmic reticulum, Golgi bodies, lysosome, and vacuoles. Now vacuoles is kind of like a storage house, so they don't have protein and they are covered with tonoplast, tomopaste. So vacuoles are covered with tomoplast. This is important. Then going on to lysosome. What is lysosome? So lysosome are the single membrane pouch-like structure found in digestive hydroxide enzyme. Worst management of cell fusion in particles, waste, and etc. Lysosome burst and does function properly. So what does lysosome does is basically like garbage collector, and it is also known as suicidal bag of the cell. And this is done in the animal cells. And they do synthesization of uh, rough, rough endoplasmic reticulum. Now, going on ahead, vacuoles. Vacuoles are like pouch, as I told you earlier. They helps in storage. basically in plant cells, and in animal cells they are there but they are not that big. In plant cells they cover up to 90% of the space, and the vacuoles are covered with tomoplast. It is their single membrane, what I was talking earlier. This is important, and vacuoles is that, right? Now centrulose is the cell division, no membrane, division of cell. Premesos is small vessels. Now division in difference in plant and animal cell. Now plant cells are usually larger because they have to do much. They are the only producer of food, and they don't have life in it. They do, but in a sense, like they don't have that much energy. Like they don't have mitochondria, right? That much. They don't produce that much energy. Uh, then cell wall present in addition to plasma membrane. So plasma membrane is there. Plasma membrane is there, and also cell wall is also there. Whereas in animal cell wall is absent, and they are smaller than plant cell. Going on ahead, plant cells have plasmodesmata present. This helps in linking the plants. We in the animal cell there is no plasmoda because animal cell doesn't link that much linking stuff, because uh, plant cells need that because they don't have that much energy to hold their own stuff, right? They need something to hold on. Next is chloroplast, which is present here, which is the kitchen, and here it is absent. Vacuoles, which are tonoplast, is there in the outer coverage, and vacuoles are large in plant cells, and permanent in plant cells. In animal cell they are small, and they are temporarily. Then centrolus absent. This is for movement. Obviously in animal cell this would be present. Centrolus present. Then in plant cell, the nucleus is at a corner, and at the middle it is your vacuoles, which is taking the most of the space. As we talked about, vacuoles take 90% of the space in the plant cells. So and the nucleus is cornered at the plant cells. The nucleus is at its center in animal cells, because here the nucleus is the brain which is controlling everything. Here nucleus doesn't need to control that much stuff. It is done by the automatically. He does, but not that much controlation is needed. Then lysosomes are there. because lysosomes is the garbage and suicidal bag, and the plant doesn't need that much here. And in the animal, it is present as animal cells produce more waste than the plant cells. So yeah, and storage material is starch grain, and storage material is glucose. Here in the plant cell, stuff is stored in starch, and here it is glucose, glycogen. Sorry. Going on ahead, cell division. Here the cell division purpose is growth. Its another purpose is repair the damaged cells. Then gamete formation. So like cell division is also done in for like to reproduce, not reproduce, but to like uh, make their population more in a sense. So the gamete formation is done by mitosis and meiosis. First is meto. Meto means two. Mitosis. And meiosis means four. So in mitosis, it goes like this, like this is there, then this is there, then this is forming here, then it becomes like this, then the whole thing separates. So this is called prophase, then promatophase, then metaphase, then anaphase, then telophase, PPMAT. Prophase, uh, prometaphase, metaphase, PPMAT, anaphase, telophase. And in the meiosis, it is something like one goes to two, then this goes to two, and this goes to two. So one is making four here. Tell me what I'm thinking of. Living tissue, single cell outmost layer of green plants, leaf, green stem, helps in fortifying of plant and prevent them from discoloration. Epidermis. Ah, where is the pen? So, epidermis dermis. This is a living tissue and it helps in fortification of plant and prevent them from drying up. Helps in fortification. If it is doing fortification, then you can obviously assume that it is at the corners. It helps in fortification uh, of plants, and at the corner it is so it prevents them from drying up. An epidermis cell produces a sticky substance called cutin. So this drying up process, the main thing which helps to plant to not dry up is cutin, which is sticky. Going on ahead, xenophytes. Xenophytes are basically when the leaves of the of leaves are like thick. Cutin is there, like cutin is the outer layer, right? So here the cutin becomes thick. So obviously like thick means in a sense it is not like a leaf, it is like straight, which is usually in cactus. Then cork is a dead thing, which is the bark, also known as PELLEM, phylum. Phylum. And cork floats obviously. An outer layer of non-green plant, it is non-green. Then parenchyma, living tissue, also help in storation. Basic fundamental tissue, thin as cell wall having intercellular space. Then this is important. Parenchyma help in storage, and like parenchyma, if it is like helps in storage, and fundamental tissue, and like parenchyma means purpose is like if parenchyma is filled with uh, like chlorophyll, then it becomes chloroplast. If it is filled with air, then it becomes aerochyma. Aaron, Arenchyma. Chlorophyll, chloroplast, air, aerochama, and if it is filled with waste, it become idoplast. So if it is filled with chlorophyll, then chloroplast in parenchyma, chlorophyll, chloroplast, air, aerochima, waste, idoplast. Going on ahead, there is meristematic tissue and permanent tissue. Meristematic tissue is something I can tell you with a plant example. Is there no brown here? Supposedly, this is the plant thing, and this is the soil, right? And the plant is growing up, and supposedly, this is the leaves. right? Now here's stuff is to learn. Now the April meristematic, which is here, APP, apical meristematic, is at the tip of the leaf to help it grow. Now inter is at the leaf and the midrib of the leaf, which is the whole thing which hold, and the stem it is joint, which is intercalary meristematic, and then there is lateral meristematic, which is at the stem. This is all meristematic. Now the permanent tissues are epidermis, cork, which we have done, epidermis, which we have done, as epidermis was the one which helps the plant to not dry up, as cutin is there, and then parenchyma, which we have, uh, I think we have to do, I think so, then chlorenchyma, then sclerenchyma. So pa- parenchyma is basically it helps the corners of the leaves to give them extra rigidity. Then scalenchyma is the only chyma which is dead. This is how produce. Sorry, how I say it. Scalerenchyma, dead, living. Believing is parenchyma. Scalenchyma is dead, and the thick wall also pigment, lysine, and yeah, lemon is immune space. Types of lumen: a narrow lumen, fibrous, and thickened lumen, cellulose. So it is like kind of like fibers kind of thing. So narrow one are called lumen, and the thickened one are called scirillus. Now uh, complex permanent tissue function is different from cell function separately at a small but also work together in single major function. Vascular system condition helps in the proportion. Types: they are small in plant as they don't need it. Plant kingdom. So thylophyta, then brophyta, then Pteridophyta, then Gymnosperm, then Angiosperm. So basically, what he's saying is in Thallophyta, there is no xylem phloem. Bromophyta, we also know xylem phloem. Petrophyta, petrified it is, so it is starting to do it, xylem and phloem. Then gymnosperms, which is in small plants, the xylem and phloem. Angiosperm is the big one which has in the big plants. So angiosperm is the one which have the xylem and phloem the most. Angiosperm is the one which is in the small plants, and then petrophyta is just starting, and broro and thallophyta doesn't have xylem and phloem. Now composition of xylem is stretchy vessels, xylem which is parenchyma which is dead, parenchyma which is living. Vessels is also dead, and everything is dead. It's just the parenchyma, xylem which is living, and like the helps. Xylem and phloem helps in water and food transportation, and they helps by like how this is a wait. This is a leaf, right? This is the midrib of leaf, right? right? Now this is going down, right? What happens is when the sun is there, sun evaporates the water which is on the leaves, and when the water get evaporate, what happens is that there is a suction kind of thing which is formed, which is your called transportation pull, which basically helps the water to come up in opposite direction of the gravity. And the xylem and phloem have cohesion and adhesion. Cohesion and adhesion. As cohesion is the water molecules stick together, which is in xylem, stick water together. And adhesion is like friction kind of thing is going on. So adhesion is the walls are sticky for water. Adhesion, sticky walls, cohesion, sticking the water itself, and transportation pull is with the help of the sun, and the water is evaporating and the suction kind of thing is happening. So this is whole how xylem and phloem works. And yeah, this is the whole plant kingdom which we have done. Now we will in next day we will do tissue.